Hi everybody, I'm Tattagavalli working as an assistant professor in the Department of Biotechnology, K.S.Rangasamy. In this, in this we are going to see about Hybridoma Technology, which is one of the most commonly used and commercialized technology. First we have to see what is monoclonal antibodies and what is antibodies. Antibodies is a protein used by the immune system to identify and neutralize foreign objects like bacteria and viruses. When our body gets infected by the viruses and bacteria, our body will produce antibodies that will protect our body from the foreign particles. Each antibody recognizes a specific antigen unique to its target. Antibody has a structure of constant region and variable region. Based upon the variable region, we can classify the antibodies into two types. One is monoclonal antibody, another one is polyclonal antibody. Polyclonal antibodies are antibodies that are derived from the different cell lines such as HeLa cells. Such as a polyclonal antibody response facilitates the location, phagocytosis and complement-mediated cell lysis of antigen. It will lyse the cell by phagocytosis, and then monoclonal antibodies have specific site in the antigens. It refers to the antibodies that are identical because they were produced by one type of immune cell, specific B cells, and then all clones of single parent cell. It all come from the single parent cell that it will recognize only one particular of antigen which is vary from the variable region. Next we will compare both polyclonal antibodies and monoclonal antibodies. Polyclonal antibodies, it will produce by large amounts and then it is non-specific. It can bind to any part of the antigens, and then coming to the monoclonal antibodies, it will produce by large amount of specifically by one cell, and then but it is not too much specific. And then in polyclonal antibodies, it recognizes multiple epitopes on a foreign particle, but in case of monoclonal antibody, it will recognize only one epitope at the foreign particles, and then it can be batch to batch variability. It will vary according to the batch, and then in monoclonal antibodies, once a hybridization is made, it is a constant and renewable source. It can be renewable from the complex of antigen and antibody complex, and all batches will be identical. It will be bind to specific antigen and antibodies. In case of monoclonal antibody, it will be very expensive and it will consume more laboratory cost to produce it. And then in case of polyclonal antibodies, it is inexpensive to produce and no high-tech technologies are required. Time scale is too short for that. Next. And then coming to the Hybridoma Technology, for producing monoclonal antibodies, Hybridoma Technology is used. It is discovered by Copeland, Milston and Jen in 1975. They are awarded for Nobel Prize for finding this Hybridoma Technology. Next. Then we have to know first what are the requirements for Hybridoma Technology. We have to clear about what is hack media and HGPRT mutated myeloma cells and primed B cells. These are the requirements for Hybridoma Technology. Hypoxanthine aminopterin thymidine kinase containing media. We are using this hack media for culturing the monoclonal antibodies to specifically identify and purify that. Then HGPRT mutated myeloma cells. Myeloma cells, in sense, it is a cancer cell which is used to produce the monoclonal antibodies. Hypoxanthine guanine phosphoryboside transferase enzyme. It is used in the pathway of de novo and salvage pathway to produce nucleotides in the mechanism of replication. We are using the mutated cells, then primed B cells. It will be derived from the spleen cells of mice, which will produce the B cells, and then it will have the ability to produce immunoglobulins to act against antigens, and it will also produce HGPRT, which is hypoxanthine guanine phosphoryboside enzyme. Then polyethylene glycol to fuse two cells, to fuse cancer cells and also the B cells. We are using polyethylene glycol here. Then these are the two pathways used for monoclonal antibody production. One is de novo pathway and another one is salvage pathway. Phosphoryboside pyrophosphate is from that aminopterin and nucleotides are produced. From this pathway, aminopterin is used as a to inhibit the pathway. Aminopterin is used and nucleotides can't be synthesized in the de novo pathway. And then in the salvage pathway, there are two types in salvage pathway. One is thymidine, one is the hypoxanthine. The nucleotides can be synthesized from the thymidine also and hypoxanthine also. For that, it needs two enzymes. One is thymidine kinase, another one is HCPRT. We are using HCPRT media. This pathway can be inhibited by mutating those genes in the myeloma cells. So, using this two pathway, we are going to produce monoclonal antibodies. Next, myeloma cells used in the hybridoma technology, in the sense cancer cells that we are using in the production of monoclonal antibodies, it won't have that HGPRT gene. In the sense, the gene will be mutated in that cells, and HAD media contains hypoxanthine. It is used for myeloma cells, for the selection of myeloma cells. Then mutated myeloma cells cannot use the hypoxanthine because it lacks HGPRT gene. Then it cannot make purines to replicate the cells. So the salvage pathway, because they are mutated in the HGPRT gene and we are using aminobutyric HAT media, it stops the action of tetrahydrofolate and cells can't make purines. So the B cells will be died. Then thymidine, it only supplies nucleotides base for the process of replication. And then after fusing the myeloma cells and B cells, we will have three possibilities. One is unfused B cells, and another one is unfused myeloma cells, and another one is the possibility of correct fusion. Possibility of correct fusion is fused B cells and myeloma cells. From this, unfused myeloma cells will die because they don't have a gene product to replicate the genome. Then normal B cells will die because it is mortal. Myeloma cells won't die because it is immortal, it is a cancer cell. Then only fused cell will die in HAT media. Immortal genes from myeloma and salvage pathway genes from normal B cells. This is the overall method we are using for monoclonal antibody production. First, we are extracting the spleen cells from the mice. For that, we are injecting the antigen into the mice. It will produce antibodies against that particular antigen. It will have HGPRT gene and immunoglobulin, and it is mortal. It can die at particular time. Then we are taking the myeloma cells, which is a cancer cell. It is lack of HGPRT gene and it can't produce immunoglobulin, and then also it is immortal because it is a cancer cell. And we are fusing those two cells using polyethylene glycol. We have 5 possibilities here. One is unfused B cell, and another one is unfused myeloma cell. We won't consider that 2 cells, and we have 3 possibilities of fused cells. One is both B cells can be fused and both myeloma cells can be fused. And then in that heterokaryotes, we are selecting only B cells and myeloma cells fusion. We are selecting that using HAT media selection, and then after culturing in the HAT media, only those possible correct fused cells only grow in the plate. Then we are taking that cells for the further steps, and we are culturing it further in microplates, and it is producing monoclonal antibodies. We are testing it using some kind of assays like ELISA. Then we are purifying that using affinity chromatography techniques. We can expand it to the clinical trials. The next. Then these are the steps I have explained in the before slide. Immunization of mice. Mice are immunized with antigen attached to an adjuvant. Adjuvant is a carrier molecule which have the antigen to carry out inside the mice. The antigen can be whole cells, membrane fragment, or complex molecule. It can be in any form of this. Mice serum is screened using various techniques such as ELISA. In the initial itself, we are doing some kind of techniques, and spleen cells is removed as a source of cell fusion. From that spleen only, we are taking the B cells for the production. And preparation of myeloma cells. Myeloma cells are immortalized cells that are capable of diving indefinitely. After preparing B cells, myeloma cells, they are fused with help of polyethylene glycol. Fusion of myeloma cells with spleen cells. It is harvested from the mice, and then myeloma cells are fused and it is cultured in HAT media. Then culturing of hybridoma cells is done in the HAT media. After hybridoma cells structured in microtiter bells in such a way that it can be grow in a good condition and it can produce the monoclonal antibodies. Group of cells is derived from the hybridoma single cell. Then screening of cells. Screening is done by the ELISA test and isolation of antibodies is done by using affinity chromatography. Thank you.